shortlisting candidates to join our team at Physiology, the company that has revolutionized the science of bodily functions. The candidate we are looking for should have a very strong work ethic, be available 24/7, have the capacity to carry oxygen in the human body, and deliver it in a timely fashion when needed. He or she should also be able to carry a heavy load of carbon dioxide and dispose of it according to waste disposal regulations, and be willing to work with human resources in regards to salary and fringe benefits. So is everybody clear on the specific requirements for the job?
Yes, absolutely.
Plasma, I have something else to add to the requirement since I'll be working very closely with a new hire.
Sure. Go ahead and tell us what's on your mind.
Well, I strongly believe that the candidate should be a team player and willing to forgo any kind of vacation time.
Does everyone agree with Mrs. Red?
I do. And we should add the requirements to the job description.
Seconded.
Okay then. I will go ahead and add it to the job requirements. Now I think it's time to review the candidates for the job. Mrs. Red, do you have your shortlist?
I do. So we have shortlisted 4 candidates for the job: hemocyanin, hemoglobin, hemoethrin, and myoglobin.
Well, I do think that judging from their previous work experience, we can rule out hemocyanin and hemoerythrin. Hemocyanin and hemoerythrin only seem to have previous experience in vertebrates. So I propose that we call hemoglobin and myoglobin for the interviewed. Are we all agreed?
Yes.
Good morning, Mr. Hemoglobin, and how are you today? Do you have any difficulty finding our offices? Please have a seat.
I am very well. Thanks for asking. No, I did not have any problem finding your offices. I'm one of those molecules that isn't afraid to ask for direction.
Good to hear, Mr. Hemoglobin. I'm Plasma, the hiring manager, and my job within the company is to make sure that the oxygen transport and bodily defense teams function smoothly. I will let my team leaders introduce themselves to you.
Good morning, Mr. Hemoglobin. Nice to meet you. I am Red Blood Cell, Mrs. Red for short. If you are hired for the position, then you'll be working very closely with me in the oxygen transport department.
It's entirely my pleasure. Ma'am, I am very much looking forward to this opportunity.
Oh my.
Hello.
Hello. I'm platelets, and my expertise mainly lies in defense and repair, but I work very closely with the other members of the team to help stop bleeding and loss of other team members.
Nice to meet you, Mr. Platelets.
Good morning, Mr. Hemoglobin. I'm White Blood Cell. You can call me Mr. White. I am trained extensively in defense functions. My team and I protect the entire organization from unexpected attacks by extracellular terrorists.
Nice to meet you, Mr. White. You have such an honorable job.
Thank you, Mr. Hemoglobin. Very kind of you to say so.
Now that we have the pleasantries taken care of, let's get down to business. Mr. Hemoglobin, please tell us a little bit about yourself to start off with.
I really don't like to talk about myself, but since you asked, I'll try and give you a sense of who I am and what I do. I'm a protein made out of chains of amino acids folded to form a globular structure with a hydrophobic, water-hating, core and hydrophilic, water-loving, exterior. I can carry oxygen and carbon dioxide, and in some cases, nitric oxide.
Mr. Hemoglobin, can you tell me a few more details about your structure?
My pleasure. Mr. Platelets. I happen to have 4 polypeptide chains made up of amino acids joined together by peptide bonds. My chains are named alpha and beta. I have 2 alpha and 2 beta chains. My primary structure can further fold up to give rise to secondary structure elements, such as alpha helixes and loops. Most proteins also have beta sheets, but I am comprised primarily of alpha helixes.
What exactly is an alpha helix, and how is it different from a beta sheet?
Good question. An alpha helix is a right-handed spiral structure that is formed due to twisting of the peptide chain. There are an average 3.6 amino acid residues per turn of the helix. It looks like a corkscrew. My alpha helix structure is stabilized by the formation of hydrogen bonds between the amino acids making up the chain. On the other hand, beta sheets are made up of amino acids joined folded to form a fan-like structure, stabilized by interstrand hydrogen bonding. Both of these elements are repetitive structures and occur many times in a protein molecule. The alpha helixes and beta sheets are joined together by loops or random coils to form a tertiary structure.
Thank you, Mr. Hemoglobin. That was very helpful.
Yes, yes, this is all very well, but what does this have to do with you carrying out your job?
Now, Mr. White, let's not get impatient. Let's give Mr. Hemoglobin a chance to explain himself.
Thank you, Mr. Platelets. I was just getting to that point. But first, I need to explain my quaternary structure to everyone. As I was saying, alpha helixes and beta sheets connected by random coils come to form a tertiary structure for a chain. As I mentioned, I have 4 chains, 2 alpha chains and 2 beta chains. When all 4 of these chains come together and are coordinated with heme in each chain, this gives rise to my quaternary structure. Each chain is called a subunit. And when all 4 of my subunits come together, then I am complete. Because I have 4 subunits, I am referred to as a tetrameric protein. And because I contain iron, I am also referred to as a metalloid protein. My quaternary structure thus contains more than one polypeptide chain that differentiates it from the tertiary structure. Just in case you're wondering, my iron can bind oxygen, and the porphyrin ring surrounding my iron ion is responsible for binding globin.
This is absolutely fascinating. What an extremely complicated structure you have.
Why, thank you, Ms. Red. I will take that as a compliment. Yes, my structure is rather complicated, and it was solved way back in 1959 by a scientist named Max Perutz, who co-shared the Nobel Prize in 1962 for elucidating my structure.
How exactly did Max Perutz discover your structure?
I'm glad you asked that question, Mr. White. He used a technique called X-ray crystallography. He first of all used various chemical solutions to make me form crystals. Once he had nice, well-formed crystals, he passed X-rays through me. The crystals scattered, diffracted the X-rays, and the points of diffraction formed a pattern on an X-ray film that was placed behind me. Using this pattern, he could make complicated calculations to determine my structure.
This is fascinating stuff, but how does your structure make you well suited for the job?
I was just coming to that part. The component of my structure responsible for binding oxygen is the iron, or heme. Since I have 4 subunits, each with its own iron, I could bind up to 4 molecules of oxygen at a time.
How does this compare with Mr. Myoglobin?
My understanding is that he can only carry 1 molecule of oxygen at a time.
Is that so?
To the best of my knowledge, sir, it appears to be true.
So where exactly does oxygen binding occur?
In the arterial blood. Because I'm oxygenated, the arterial blood appears a brighter shade of red due to the oxygen binding to my red iron groups.
So it is you who imparts color to the red blood cells.
Yes, ma'am, I believe so.
Can you explain how exactly the oxygen binds to your heme?
Absolutely. The fact of the matter is that oxygen is not very soluble in an aqueous solution such as blood, so it needs to bind to me in order to be transported. Each of my iron ions can bind 1 oxygen molecule, so I can carry 4 oxygen molecules at a time. This is called my oxygen carrying capacity. The reason why my oxygen binding to me is very efficient is because of the process called cooperative binding.
So what exactly is cooperative binding?
Very simple. It means that once 1 oxygen molecule has bound to me, I can change shape, undergoing a conformational change rapidly in order for the 2nd, 3rd, or 4th molecules of oxygen to bind to me more efficiently. Changes in the structure of one of my subunits are translated into structural changes in adjacent subunits. My structure can change with the binding of oxygen from a low affinity state to a high affinity state, which is the relaxed state.
How does that work?
When my iron is oxygenated, it moves closer to the porphyrin ring. As a result, the histidine residue to which the iron atom is attached is drawn closer to the heme group. This movement of histidine residue then shifts the position of other amino acids that are near it, and the structure of the interface, where the four subunits interact, is altered, which leads to other structural changes. What this means is that when a single hem group in me becomes oxygenated, my entire protein changes its shape. In my new shape, it is easier for the other three hem groups to become oxygenated. Thus, the binding of one molecule of oxygen enhances my ability to bind more oxygen molecules. This property is known as cooperative binding. I can bind oxygen cooperatively so that in places of the body with high oxygen concentrations, such as in your lungs, I can take up oxygen. The cooperative binding of oxygen enables me to deliver almost 1.7 times as much oxygen than it would be if the sites were independent. My regulation of oxygen dramatically increases my physiological oxygen carrying capacity. In the tissues where oxygen concentration is low, I can give up oxygen also in a cooperative manner, thus releasing oxygen efficiently into the tissues.
Okay, so I get that you can efficiently carry oxygen, but how do you release oxygen, carry carbon dioxide, and release it into the lungs for disposal?
Excellent question. I mean, this can be explained by a phenomenon called the Bohr effect, where hydrogen ions and carbon dioxide help to promote the release of oxygen.
It all sounds very complicated to me.
Not at all, ma'am. It's actually pretty simple. My oxygen affinity decreases, the pH decreases from the value of 7.4. You know, the pH decreases mean that the hydrogen ion concentration becomes higher. Thus, as I move into regions of low pH, I tend to release oxygen. In addition, I respond to carbon dioxide with a decrease in oxygen affinity, which helps the release of oxygen in tissues with a high carbon dioxide concentration. This type of regulation of oxygen binding by hydrogen ions and carbon dioxide is called the Bohr effect, after Christian Bohr, who described the phenomenon in 1904. I am carried with bond carbon dioxide and hydrogen ions in the blood back to the lungs. In the lungs, I release the hydrogen ions and the carbon dioxide and rebuild oxygen. Thus, I help to transport hydrogen ions and carbon dioxide in addition to transporting oxygen.
Mr. Hemoglobin, thank you very much for your time. The conversation with you has been extremely interesting. We will let you know the results of the interview at a later date.
Thank you all for having me. I look forward to working with you all in the future.
Well, ladies and gentlemen, please fill up your evaluations and I will call on the next candidate. Janet, please send in Mr. Myoglobin. Mr. Myoglobin, I'm Plasma. I'm very happy to see you. Please, take a seat.
Thank you so much.
Mr. Myoglobin, let me introduce you to Miss Red, Mr. White, and platelets.
Pleased to meet you all.
Mr. Myoglobin, I'm the division chief for the Oxygen Transport Group. Can you tell me in a few words why you would be a suitable candidate for our team?
Certainly, Miss Red. I'm a globular monomeric protein found primarily in the muscles, and I can bind oxygen molecules delivered by hemoglobin. I am capable of binding a single oxygen molecule.
I see. So you're not primarily found in the blood.
No, ma'am.
Thank you. Can you tell me a little bit about your structure?
I contain a single polypeptide chain and am built primarily of alpha helices and random turns. I contain a heme group made up of a protoporphyrin and a central iron atom that allows me to reversibly bind oxygen. My interior consists almost entirely of hydrophobic or water-hating residues. The only charged residues that I have inside of me are the two histidines that play a vital role in the binding of oxygen.
So what is the mechanism by which you can bind oxygen?
It involves a bit of complicated chemistry. My iron can bind to 4 nitrogen atoms in the center of the protoporphyrin ring, leaving 2 free bonding sites. One site bonds to the histidine, the other is free to bind with oxygen. This histidine prevents other larger molecules from entering the oxygen binding site and also makes the reaction reversible.
So what you're saying is that the oxygen carrying capacity is 1 molecule of oxygen per molecule of myoglobin?
That's correct.
How does your structure differ from that of Mr. Hemoglobin?
Hemoglobin is a tetramer compound of 2 each of subunits alpha and beta. I'm a monomer, so I don't have a quaternary structure at all.
So in terms of function, how do you differ from Mr. Hemoglobin?
Well, sir, I can bind oxygen much more tightly than does hemoglobin. That's why I'm well suited for binding oxygen in the muscle. Additionally, I can act as a backup system for hemoglobin under extreme conditions like exertion or exercise. When hemoglobin can't supply enough oxygen to the muscle, I can take over because I am able to store oxygen. Since I hold on to oxygen more tightly, letting go of it isn't easy. As a result, I'm able to store oxygen reserves in the muscles.
So what you are basically saying is that you don't release oxygen into the blood. But why?
I don't release oxygen into the blood because I have a higher affinity for oxygen than hemoglobin. Furthermore, the partial pressure of oxygen in mitochondria is much lower than blood, so that when I do give up my oxygen, the concentration gradient favors delivery to mitochondria rather than blood. This property can be illustrated with the help of graphs that I brought to show all of you.
Very nice.
Thank you.
Incredible.
Oh, now I understand. So you would be more useful in the environment of the skeletal muscle when hemoglobin is under excessive pressure.
That's correct, ma'am. We like to work as a team, hemoglobin and I. My buddy hemoglobin carries the oxygen from the bloodstream, then passes some of it to me in the muscle cells for storage until it is needed in stressful situations like exercise.
So as I mentioned, I'm the division chief for the oxygen transport group. However, my deputy, Mr. Muscles, is responsible for recruitment to the oxygen and exercise branch of the division. Let me see if he is available to join us for this interview.
Sounds like a great idea.
Mr. Muscles, can you come in? Mr. Muscles, do you have any specific questions for the candidate, since this is your area of expertise?
Absolutely. Thank you, Mrs. Red, for calling me. Now, Mr. Myoglobin, it is my understanding that you function primarily in the skeletal muscles rather than in the blood. Why is this?
Well, as I was explaining before, the blood needs to be able to lose oxygen as well as capture. The whole point is to take oxygen into the lungs, and then the lungs, and then release it throughout the body. Blood that is really good at only capturing oxygen would be an oxygen trap. If I were to be present in the blood, I would not want to let go of the oxygen because I have a high affinity for it. That is why I take oxygen from hemoglobin and store it in the muscle for future use.
That's a good answer. I have one last question for Mr. Myoglobin. Why do deep diving in sea animals have higher concentrations of myoglobin?
Great question. Deep diving sea mammals take long underwater dives. In order to have enough oxygen stored to allow them to do this, they need a much higher concentration of meat. Usually when proteins are highly concentrated in one place, they start to clump together, which stops them from functioning properly. In the case of deep diving sea mammals, they have evolved variants of myoglobin that are less likely to clump together because they have a highly charged surface that allows them to remain at high concentrations without a loss of function.
Good answer. Thank you for your time, Mr. Myoglobin. We will let you know our decision at a future date.
Thank you very much, everyone. I look forward to hearing from you.
Now, ladies and gentlemen, we have the very difficult task of choosing whom to hire for the job, Mr. Hemoglobin or Mr. Myoglobin.
Well, it's not difficult at all, Mr. Plasma. I choose hemoglobin for my oxygen transport division. He is more valuable to me than myoglobin.
But Mrs. Red, my branch cannot function properly without Mr. Myoglobin, if you know what I mean.
So, who do we hire?
We hire them both. Mr. Hemoglobin will take care of oxygen transport in the blood, while Mr. Myoglobin will take care of oxygen storage and release in the muscle. Does that sound reasonable to everyone?
Yes.
Yes.
Great
Great
decision. Fantastic.
Okay then. Let's get all the paperwork done and invite both Mr. Hemoglobin and Mr. Myoglobin to join the Physiology team.
Ring, 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 ring.
Well, hi there, Mr. Hemoglobin. I am proud to announce and to congratulate you on getting the job here at Physiology.
Thank you so much. It was such an easy interview, so I'm not surprised. Thank you.
We look forward to you being a team player. Thank you.
Go.
Ring, ring, ring.
Hello?
Hey, is this Myoglobin?
Yes, it is.
Guess who? It's Mr. Muscles.
Yay.
Anyways, girl, been trying to reach you, you know. I got good news and I got bad news.
What's
Which
the good
one do you
news?
want first?
Good news.
The good news is you got the job. Welcome
Yes.
abroad. You ready for the bad news?
I guess.
I spilled half my protein shake this morning.
Oh.
I ain't getting a full pump right now.
I'm so sorry.
Anyways, welcome to the team.
I'll bring you some protein.
I like her already.
Just
get a nap.
do the Mr. Muscle voice again. You just like come up with that on the spot.
But Mrs. Red. My bridge. I was trying to sound like I'm full of testosterone.
Yeah, yeah, very good
Okay.
job.
Okay.
This
Great.
last scene is really
I talk to myself.
Just like
I'll be like,
Slide to the other side.
Muscle. Be like, fine, platelets. Well, I
okay.
don't know, whatever I say.
You should drink and then reappear. Like, right away.
Yeah, I'll just be like, like that. That's all I do.
Do it. Go for it.
Good morning, Mr. Hemoglobin, and how are you today? Did you have any difficulty finding our offices? Please have a seat. Wait, can we read?
And then I'll just say, congratulations! We have accepted you for the job, Mr. Hemoglobin. I look forward to you being a team player here at Physiology.
Can I make a honk noise? I'm really good at making a honk noise.
What does the honk noise have to do with it?
Well, it's just like, it could be like your phone ringing,
You are really good.
you know?
What does that have to do with
Okay,
it?
I'll just make a ring
The
noise.
creative license. Go for
Just
it.
make
Okay.
a ring. I'll just do ring.
Okay.
Go ahead. Ready?
I can be a ventriloquist. Ring, ring! Ring, ring! I'll just do that.
Yeah, he can just do it.
Okay.